Vamos a usar los micrófonos porque vamos a transmitirlo en línea. Estamos esperando que entren todos. Entonces, tú nos dices, Ángel. ¿Listo? Okay. So we are already broadcasting online. So thank you, David, for being here. I'm going to introduce David with a brief bio of his career. David is a professor at Newcastle University. He's a civil engineering and has a master's degree in environmental engineering by University of British Columbia and a PhD in environmental engineering by University of Arizona. He has researcher and/or professor at the University of Arizona and the University of Kansas, and since 1999, he has been professor at Newcastle University in the UK. In the last decades, he has become one of the experts on antimicrobial antibiotic resistance, emphasizing several works understanding the role of wastewater treatment plants in controlling such problems. In this sense, he has been a technical advisor or responsible for several governments and international organizations like the US Presidential Council on Antibiotic Resistance, co-leader, author of reports for the World Health Organization, and more recently, member of the technical group on antimicrobial resistance at the United Nations. No less important, he is author of more than 150 indexes international scientific papers with an age index of 52 and close of 10,000 of citation of his works. So thank you, David, for being here, and thank you, our university, for funding this visit, and the floor is yours.
Can everyone hear me okay? Okay. I'm sorry, I'd love to be able to give this presentation in Spanish. I tried to do it once, and 10 minutes into the talk they said, please speak English. So I aspire to one time in my life give a talk in Spanish, but it won't be today for everyone's benefit. I'm going to talk, my talk is going to be quite general, and it's really to provide overall guidance and discussion of the types of things people are doing at different levels within intergovernmental agencies relative to AMR. And the talk's called One Health Thinking and Integrated Surveillance to Prevent AMR. The first part of the talk, there we are, okay. First part of the talk, I'm really going to talk in very general terms. I'm going to talk about the development of a report called Bracing for Superbugs, which we were responsible for preparing, that came out about 6 months ago. And it's really a background to argue in favor of including environmental surveillance as part of solving AMR as a global problem. And I'll spend about the first 15 to 20 minutes talking about this, because this is, I think, among the most, this is really probably more important than the technical side of the talk, because I think it's information that people who work on the engineering side need to know about relative to having greater influence in terms of the work that they perform. The second part is I'm going to give you a couple of examples of what integrated surveillance looks like. And I'll define what integrated surveillance is in the first part.
People online doesn't hear you.
Okay. Am I talking? Oh, okay. Whoa. Okay. That's funny. Okay. I've got it. Normally there's no problems whatsoever in hearing me talk, but obviously this mic is not as good for me as others. Okay, so what I basically going to, first part is going to be background to do with Bracing for Superbugs reports and its recommendations on integrated surveillance, and the second half is providing some examples of what integrated surveillance looks like. And a lot of the goal in this presentation is to try and help people who do environmental AMR, antimicrobial resistance, have greater influence relative to the work that they perform. I realize that not everybody in this audience has got AMR backgrounds, so I'm going to talk about this from basics to somewhat more complicated, but generally speaking, this talk is not that complicated. It's not that scientific. The report that came out here is, this is the book, is the front of it. It's Bracing for Superbugs, Strengthening the Environmental Action into One Health Response to Antimicrobial Resistance. And it was commissioned by the United Nations Environment Program to try and increase the case for including environmental factors in World Health Organization policy to do with antimicrobial resistance prevention. The report was, as you'll see, was highly successful. I was one of the lead authors on this, but there was over 60 authors in different contributing parts. And I know that a number of people who are not within this room, but online, are people who are also contributors relative to the report. So, I'm firstly going to really simply define what antimicrobial is and why it occurs. Now this is, usually I would take about 20 slides, but I'm going to use one. So this slide is going to summarize a lot of things in a small space. Basically what antimicrobial resistance is when a bacteria or protozoa or any microorganism is exposed to a chemical that causes it stress. A chemical could be an antibiotic, it could be any other pollutant. Causes it stress. The majority of the organisms die based on that stress. But as a result, there are some organisms that don't die. And what you've functionally done is created a selection process that's selecting for slightly stronger organisms. And so this is happening predominantly as a consequence, partially, of the fact that we use antibiotics more and more and more, and we also use them inappropriately in ways that maybe they weren't intended to be used. So what happens is the organisms, they compete, they go into the microbial ecological world, and these organisms that are resistant become dominant. Now, the reason why this is a problem, it's obviously a problem intrinsically, but it's also a problem because this is happening absolutely everywhere all the time. It's happening in medicine, it's happening in veterinary medicine, it's happening on the land, in water, it's happening everywhere. The problem is because all of these places where antimicrobial resistance is getting selected is connected by water and air, that you can't tell which pathways are the dominant pathways. So I would be willing to guess, so I won't put a number, is that I would say probably 60 to 70% of the AMR cases you find at a hospital are almost certainly driven by environmental factors. In other words, they're acquired resistance that's been acquired in the community that the patient has taken it to the hospital. And the problem is that we have had a terrible time convincing people in the medical world, in the veterinary world less so, that this is critically important. And that's what this report was to do. It was to try and raise the awareness amongst the chief scientists within the World Health Organization and the United Nations and other governments into the role of the environment. And as I said, we were successful. And one of our conclusions is One Health, which is holistically considering humans, animals, plants, and the environment as intrinsically interlinked. In other words, they're interlinked by nature. And that if you try and prevent something like antimicrobial resistance without considering those interlinks, you're not going to solve the problem. So the positive news is, as a consequence of this report, the United Nations went from tripartite in human health protection to quadripartite in human health protection. This was changed about a year ago. And what quadripartite means, four, and that means animals, humans, plants, and the environment. Before it was animal, humans, and plants. So this report's been quite influential. It's changed a whole lot of thinking, both at the highest levels and also on an operational level. So let's think about why it took so long, considering I was studying environmental AMR in 2003. And I couldn't convince anybody that what I was doing was relevant in medicine. And yet I was positive it was. The problem is what I showed in my original slide. You've got overlapping local pathways that are all overlapping. And in fact, you can't tell which direction of flow the resistance is occurring. If you find something everywhere, you don't know where it's coming from because it's everywhere. So we know with some confidence now that the environment mediates much of the spread, usually by water. And what our goal is, by now recognizing this interconnectedness, is that we can identify the key sources using more and more sophisticated methods, but also then slow the pathways. So by fixing the environment, we will fix medical AMR and veterinary AMR. That's the goal. Now, one of the other problems, and this is a really big problem that I think people on the engineering and the environmental science side don't quite understand, I'm sorry, is what the risks of AMR actually are. And this is something that I don't think people completely understand. So, and first, antimicrobial resistance is the ability of a pathogen, an organism that's going to make you sick, to not be treatable. So they've developed some genetic trait or protein, it's acquired a protein that makes it resistant to therapeutic treatment. Now, there's two problems, two ways that's a risk. One is, well, the risk is you might ingest a drug-resistant pathogen. So it's a live organism that's got resistance capacity that when you treat with that drug, it won't work. The biggest consequence of that is you die. And I think we've thought for a long time is that's the most important risk. Now, it is an important risk. It's actually not the most important economic risk. The economic risk is you getting a disease that makes it such that you have to stay in hospital for 3 times the stay. That's a bigger cost, and but that's a different talk. However, the first one is you ingest a viable resistant pathogen. The second risk is more subtle, and that is as we pollute the environment, as we use antimicrobials, as we release waste, we're slowly increasing the genetic potential of the environment to be resistant. We're increasing the gene pool, we're creating selection and evolution of new types of genes, and those are just sitting around in the background. And so what we've functionally done is we've accelerated evolution by polluting the environment. So the first risk is you die. The second risk is that we have increased the probability of any pathogen becoming resistant, because the gene pool that could make an organism resistant has increased in nature. So when you think about that within the context of integrated surveillance, and now how do we measure resistance on a broader scale? The question is, what do we want to measure? What are the things we want to know? This is an organism, this is probably genetic potential. So they're two very different things. So let's think about this. I've got 4 things you want to measure, and I prioritized in the order in which I say that feel they're important. First is, we want to measure antimicrobial resistant pathogens. These are living organisms that are drug resistant. You can't do that genetically. Doing metagenomes or all that stuff, it doesn't tell you anything about this. You have to do classical microbiology and grow organisms on plates. So that's the biggest issue, and that's going to tell you the potential of ingesting that AMR pathogen that's maybe untreatable and you might die from. The second most important thing is viable resistant organisms that are in nature that aren't pathogenic. So these are the ones that don't cause disease, but they're active organisms that are potentially going to exchange the genetic potential to a pathogen. So we're still talking about viable organisms, living organisms. Now, this is where people who do environmental research usually do most of their research. They quantify the resistance genes and mobile elements that carry resistance genes, and they quantify those genes. They don't know where those genes are in. They don't know whether they're in an organism, whether they're present as a free genetic material. We don't know what they are, except we know they're there. The thing is that an AMR gene has never killed anybody. It's just a reflection of the genetic potential for resistance. The only way that resistance gene is going to have any real consequential effect on health is if it gets into that organism and eventually gets into that organism. So the problem is, we as environmental researchers, we study this. The people who are dealing with disease and medical systems are studying this. And that's the reason why people who do environmental research don't have much influence. We're not providing the medical and veterinary people the right information. So what we've got to do, we know from experience we can do this really well, AMR genes. But we've got to be able to bring it into that context, in a resistant pathogen context, for people in the medical and veterinary world to take what we do seriously. And so, the other thing we want to know is we want to measure the chemicals. We want to get a sense of the different concentrations and types of chemicals in the environment that might cause stress. that might make our organisms more resistant. Okay, now I'm going to talk a little bit more generally about sanitation and some of the conclusions from the Bracing for Superbugs study. And this is a map we generated in some work I did for the World Economic Forum. We were commissioned in about 2020. This is a really useful report that was basically generated for the purpose of trying to quantify the economic consequences of waterborne AMR. So, and in fact, we monetized how much it costs, how much death costs, how much staying in the hospital costs, and then how does that fit into the economies of different countries. And through that, we then projected, based on a whole series of characteristics of countries and their economies, which countries are going to be more susceptible to AMR as a future problem. And as it turned out, it was really educational for me. I was on this as an expert on the AMR side, but I worked with some really great economists that taught me a lot about the economics of anything. But this map, the basic presumption in all of the stuff we do is that the underpinning factor related to anything to do with disease transmission is related to waste releases into the environment. And this map shows you the quality of water sanitation and hygiene around the world. The colors reflect the deepness of the color tells you how poor that particular area is relative to the quality of sanitation. So you see the worst part of the world is sub-Saharan Africa. You can see South America, including Mexico and Latin America, and Southeast Asia. They also have moderately low levels of water sanitation and hygiene. This is as defined by the definitions of the World Health Organization. So what we wanted to do is say, okay, if our hypothesis is that this is the primary driver of infectious disease spread, including AMR, how does the projected 10 years from now in waterborne AMR look like? And I want you to look at the color of Brazil. Brazil's a good one to look at, and also look at India. So in theory, if sanitation is the only driver of AMR, the colors shouldn't change. So where sanitation is poor, you should expect AMR to be poor. And look at what happens with Brazil. It gets darker. India gets black. So these are parts of the world, are places, based on looking at all the economic and other factors within that particular economy, are places where there's much greater possibility of higher levels of AMR in the future. And it's a whole different talk to talk about all the whys. But what's really, in a very general sense, it says that although sanitation underpins, or poor sanitation underpins AMR as a problem, there are other factors in some places that make it worse, and other factors in some places that make it better. So it depends upon other things like climate, population densities, and other factors. Now, the one thing that's odd to me, and I was talking with Julian about this, is Mexico is really light. And I think the reason why Mexico is light relative to AMR is there isn't any data. It's actually not because it's light. I would say it's probably more like a place like Colombia or a place like that relative to the potential. But the bottom line is some parts of the world we simply couldn't get any data, and there wasn't enough work being done to be able to allow us to answer the questions. Now, I know people are doing work, but it's not being recognized to the extent that it should. Now, how does this translate into actual resistance in individuals that live in a place? And this is this figure. I hope you can see it. It's fairly small print, but that's South America, and each of that's Africa, that's Asia, Europe. So this box, the position of the boxes here, tells you the relative number of resistance genes in the gut of people who live in that place on average. This is based on the Global Sewage Survey that was done about 6 years ago, and this has been updated recently. There's two things the few things really important to note. Europe has got the lowest per capita, It's got the most advanced wastewater infrastructure in the world. There's very few places in Europe that doesn't have some sort of advanced wastewater treatment, wastewater management. And that directly translates into this. And this is the type of evidence that the UN was now taking relative to why we have to be quadripartite rather than tripartite. The interesting thing is South America doesn't look very good. But what's interesting about South America is the size of the error bar. It's huge. Well, South America is a really big place with extremely diverse levels of sanitation, extremely diverse levels of agriculture and other factors. But it says that this part of the world is only second to sub-Saharan Africa for problems prospectively. Well, this is actually existing data. This is not projected data. This is actual data. So the levels of AMR, particularly in some parts of South America rather than Central America, have got very, very high levels. I think the highest we found was in Peru. So this is what we're facing now. This is the background. So this is the one data slide from all the things I've already told you that I'm going to show from the report. This is from going back to the Bracing the Superbugs report. So after doing all this analysis and looking at different settings and economics and all that, we came up with 4 potential parts of, in quotes, the way we are as a one health world where we have to worry about AMR. First is we have to worry about it relative to human waste. We see very strong correlations between the level and quality of water and wastewater treatment and the levels of AMR in the associated society. And it is the dominant factor. That factor then is impacted by other factors. And what we called them in the report were economic drivers. And in fact, it's the economic driver, but it's also what's called the value chain within a particular sector. And I'll explain what that means in a second. So and the 3 drivers were what we do within our healthcare systems, what we do within agriculture and food production, and what we do within pharmaceuticals and chemical manufacturing. So when we talk about that, healthcare, for example, we're not talking just about the hospital. We're talking about the pathway by which that hospital, that drug, for example, gets used in the hospital. Now that hospital, it's got the drug has got to be made somewhere. It's going to have an impact there. It's then going to be shipped. It's going to have some impact during shipping. The value chain is the whole pathway of phenomena from the point of original manufacture, for example, to the actual point of use. So this was something else that was new in this report. It was saying, okay, we've got antimicrobial manufacturing facilities, and say, for example, in India, because there's many there. For years, people have assumed that AMR is near a manufacturing facility is the predominant problem place is actually at the manufacturing facility. It's actually more than that. It's the whole pathway through which that manufacturing process influences AMR, which is originally at sources to do with the ingredients that are used to make antibiotics all the way through to the point of use. And that's what the value chain is. And so what we've said is you've got to have a view of healthcare or agriculture or pharmaceuticals, not just by treating the waste. You've got to think about more than that. It's got to be the whole process that leads to the waste. And one of the major recommendations in the report was prevention. We want to prevent the possibility of AMR getting into the environment. Once AMR gets into the environment, there's not much we can do about it. It's too late. So we want to prevent, and the only way we can know where we've got problems is by integrated surveillance. Okay, so what we want in integrated surveillance, and this is the primary recommendation, was that we want to integrate surveillance to identify and prioritize key points of intervention. We want to identify science-informed hotspots. We don't want to just use our gut feeling. We want to have explicit data to define where we need to be acting. That requires immediate action, and that varies drastically from place to place. And therefore, local activity really becomes important. We must co-develop solutions. In other words, if we're doing work that's affecting health, we've got to get people related to the healthcare world involved in what we're doing. We need to co-create how and what we should be measuring to help people make those decisions. And finally, we know that most of the solutions have to be solved at local levels. And we've got to source track and identify places where we should act on a very specific level. So what I'm going to do is give you 3 simple examples. They are all relatively simple, of what we mean by integrated surveillance of the type that we need to do to be able to try and solve AMR as a global problem relative to health, veterinary medicine, crop production, and all the different elements of One Health. So the first example is actually work we did many, many years ago. And the methods that were used for this, for this study were very primitive compared to what people can do now. All we had was 30 qPCR probes and primers. We didn't have metagenomics. We didn't have a lot of the fancy methods that people use routinely, just because there wasn't the methods available at an economic level. At the time, I was involved in a study looking at metal pollution along the Almendares River, which is just is on the west side of Havana. And it was a very interesting catchment because it had suburban areas, it had a zoo, it had antimicrobial manufacturing facilities, particularly ampicillin. It went through a wastewater treatment plant that was being impacted by the manufacturing facilities. We had a big landfill where the leachates weren't controlled, and then we had a very densely populated mixed urban environment. So we had all these different factors. And the question is, well, okay, you've got a problem. You've got AMR in the river. What do you do about it? Because you've got all these possible ways and reasons why you could have that explain AMR in that river. So what we did was in our panel of 30 resistance genes, which you would now use metagenomes in microbiology, we used our qPCR genes. We also did microbiological culturing, but I'm not going to talk about that. And what we found was, as we went from upstream to downstream, and I'm only using 3 examples of antibiotic classes. This is beta-lactams, this is erythromycin and macrolides, and this is tetracycline. And the first thing that you note is these lines don't look the same. This one is like this one goes, oops, sorry. This line goes up and then it stays level. This one goes down, then goes way up and then goes down. So the first thing you know is antimicrobial resistance in that river is not the same in different places. So you don't have one solution. There's got to be a bunch of solutions to really solve this problem because you've got different resistance from different sources. So what we then did once we had this data, we went on the ground and looked at the land use. And we found that here there was a landfill and there was a particular point where leachates were going to the river without treatment. This explains tetracycline resistance because metal is co-carried on plasmids that are associated with tetracycline resistance and metal resistance are on the same plasmids often. This was clearly the source where the pharmaceutical wastes were going into the river. The main manufacturing waste, the antibiotic, were beta-lactam antibiotics like amoxicillin and ampicillin, and beta-lactam resistance associated with amoxicillin and ampicillin. And in this case, this bump, this is basically due to leaking septic tanks. So now we had a bit of an idea based on land use and the different types of resistance we found in the river, we were able to come up with solutions. And these were acted. So we put in leachate, we brought this to the government in Cuba. Our colleagues in Cuba did, not myself. And they showed this data to the government, and they said, yeah, we understand that because it's quite simple. It doesn't take a rocket scientist to understand this, and it's real and legitimate. So what they did was they put in a leachate treatment facility at the landfill. They blocked all water flow from the landfill into the river and directed it into the catchment on the other side of the river. So they then looked at what was happening with the factories, and they put in higher levels of management relative to the production facilities and what was being allowed to be released. We never really, to my understanding, this was never solved, the leaky septic tanks in the poor areas. But the bottom line is that once you provide evidence that's clear and a pathway and mechanism that's explainable, then people do act. And in this case, they acted and they cleaned up the landfill, which desperately was important for various other reasons. And we came up with solutions. And so what this basically is a very simple version of integrated surveillance. We monitored for the purpose of trying to characterize different types of sources. And when we did that characterization, we could go to the government and say, this is a problem, this isn't a problem, this is a problem. And then they responded and acted. Okay, my next example, the results aren't so clear. And this is work that we've just finished, and we still haven't completed all the analysis. And this work isn't published yet. We're hoping to publish in about 2 months. So what we were interested in basically AMR and zoonotic infectious disease in places where people are very, very, very poor. In this case, it was in slums or informal colonies in Dhaka and Bangladesh. So what we did was with local physicians in Bangladesh and Dhaka, we looked at relationships between AMR and infant feces, water sources, and wastewater, sewage that basically runs along the streets in Dhaka. And then we also wanted to ask the question, does malnutrition influence AMR? In other words, we're dealing with people who are very poor, they're undernourished, with extreme malnutrition in some cases. And is there higher AMR in these people than we find in other places? And then how can we then go about solving malnutrition and AMR at the same time? So as you'll see, we weren't completely successful here. But it shows you the principle. So we worked with the Dhaka International Center for Diarrheal Research, Disease Research. They're one of the top research institutions in the world in this topic. So we had physicians involved. So they were out there working with the patients, all anonymized. We, as in my group, and my colleagues in the Chinese Academy of Science, did resistomes and microbiomes. The resistomes were using high throughput qPCR. The microbiomes was done by 16S Illumina sequencing. And then we collected water and sewage samples, and we collected samples from fecal from 71 individuals. This, from a medical perspective, this is a tiny study. This is a really small study, but it shows the principle at least. And then we looked at over 200 nutritional metrics of those individuals, the patients. And I've only got 2 slides for data, but I wanted to show you just the type of thing that we've got. A couple of interesting things. First, this is basically a PCA-like plot. That's fecal matter. That's sewage and drinking water. They don't overlap at all. This is looking at the relative diversity. This is based on microbiome data. That's fecal matter. This is water and sewage. So in terms of water and sewage, the biodiversity is much higher than is in the fecal matter. But based on the composition of the community, they're completely different. So when you look at the individual groups of organisms that are present in the fecal matter versus the water and wastewater, we also find they're very dissimilar. I'm not really going to highlight them. It's just there's too much data there to really see much. The only thing that's interesting here, and it sort of tells us that maybe our microbiome data is logical. This is the group of bacteria that you find in breastfed infants. So the fact that this particular type of group of organisms is highly elevated in the infants suggests that our microbiome data is probably reflecting some truth about the population that we're assessing. Now, when it comes to AMR, we see similar sorts of truths. I just want to have you look at the yellow boxes and the purple boxes. Purple boxes are humans, are the infants, and the yellow ones are sewage. And what you see is, in every case, except for macrolides and tetracycline, you find higher numbers of resistance genes per sample volume in the environment than you see in the patients, in the infants' fecal matter. The only 2 where we see higher in the infants are macrolides and in tetracycline. Now that to me is incredibly important. And the reason is the predominant drugs that are used therapeutically in those settings are erythromycin and tetracycline. Macrolide, erythromycin is a macrolide. So what we're seeing here is when we look at what's the individuals who are living in this environment, the resistance in their gut, it's not anything to do with the environment. Now, it's probably influenced a little but not within the subtlety that we could gather from the data we had. So it said that really even though there's diversity of whole different types of resistance, the quality of the environment in which these infants live is poor, that the actual elevated levels of resistance in their gut is based on the drugs that they've been taking rather than the environment in which they live. Now, the mistake we made in this study was that we didn't include food. And we're now doing a study in India where we're looking also at informal communities where we've included food. And in food, we do find relationships. So what we think is the two factors that are infecting individual health, infants, are the drugs that they take and the food that they eat. And hopefully we'll be able to tell you a nice story in about 6 months about the food side. But once again, this is an integrated study. We co-developed the study with medical people in the field, and then we provided them the data that they needed to be able to use for their own work. Okay, my final example, and I'm going to speed up a little bit because I want to be finished before 1:45. This is something that I think some people here will have done, is wastewater surveillance of AMR for health protection. And it's work that we did right at the end of the pandemic, and it was a spin-off of work that we were doing to do with COVID. We were one of the primary groups in the UK that were doing COVID surveillance using wastewater, and we basically started doing it before there was cases in the UK. And the principle behind this is that you as an individual, you've got disease in you, you take drugs, you do all the different things. All of what you do is mirrored by what comes out in your urine and your feces. So that then gets in the wastewater, and then as it moves through the wastewater system, we can calculate some sort of marker, usually genetic, of COVID, for example, and you can normalize that to the number of people from which it came, and it tells you something about the prevalence of a particular disease at the community scale. You then combine that with data from the public health people, and you end up with some sort of epidemiological model that allows you to correlate what you measure in the wastewater and what you find in actual cases. So this is however, when we were getting at the end of the pandemic, the UK government wanted us to look at things other than COVID. So they said, here's a nice amount of money. Can you try and do the same type of study that you've done for COVID, but do it for AMR instead? And that's what this is. It was done in Newcastle, which is where I live and we worked. We actually had 17 stations that we were monitoring in sewer lines that were set up during the pandemic. This particular study on AMR, we used 8 of them, and I want to highlight just one, and that's number 9. It's right here. And that was one that was just downstream of a hospital. And this is, once again, this is being co-developed with the Health Security Agency within the UK. So what we saw was, not surprisingly, we saw things that were different, that the hospital differed. Now what's interesting is this is just culturing data for E. coli and E. coli that are resistant to beta-lactams, extended spectrum beta-lactams. And so when you looked at just the organisms, just E. coli, which is in everybody's gut, we didn't see differences between the hospital and the community. But if you look at the ones that are resistant, basically you see higher levels that are coming out of the hospital. We did it with chemicals at the same time. And this is our hospital. We were monitoring 108 chemicals, and 32 of which were antibiotics. That's our hospital. These are our various different community settings. So chemicals we can use from wastewater for health monitoring. This is looking at mobile genetic elements. These are these cassettes, gene cassettes that allow transmission to occur that carry resistance from one place to another. So once again, this is our hospital. This is the number of genetic elements per cell that we find in the wastewater. We also did some background at another city in Wales. And these are our 2 hospitals, the hospital in Wales, the hospital in Newcastle, and these are our community sites. So what's happening in the hospital is quite different than the rest of the community. And it's what it amounts to being is highly transmittable resistance in the sewage that's coming from the hospital. It doesn't mean people are going to get unwell because they're not going to drink the sewage. But if you were in an environment that didn't have wastewater treatment plants, that's a very serious problem, and it's part of the reason why we think the levels of resistance in places that don't have sanitation have so much resistance. And this is just another slide that shows the same sort of thing, except this is AMR diversity. Once again, these are all community sites. That's our two samples from the hospital in Newcastle. Those are two samples from the hospital in Wales. We also can find specific markers that are very particular to places where there's hospitals. We also can find particular markers to do with other types of industries, but I'm not going to talk about that here. However, the bottom line from this, and this is where this becomes really helpful, is that we were doing this in loose association with the UK Health Security Agency. However, we went to the local UK National Health Service people, the trust in the Newcastle and Durham area where we live, and they thought this data was amazing. However, what we found valuable was they said, well, you're just not making nearly as, this is what we need to know. These are things that we need answers, questions. And there were questions that we'd never even thought about that they thought were useful. So we created enthusiasm. We had loose association with the healthcare people, but then we showed them specific data that they felt was immediately valuable. And we provided it in a form they could use. We provided them culturing organisms, we provided them genetic markers, and we provided information to do with chemicals that they use through wastewater. So they came back to us and said, okay, well, we can use this as an early warning system for any sort of disease, and that's sort of the basis of wastewater monitoring. However, what they were really interested in was quantifying chemical use and behavior at the community scale. So we as people who are given prescriptions, we take antibiotics or other drugs, but the doctors don't know whether we use them or not. So what they're using this data now for is giving them an idea of whether where people are using drugs, particularly drugs to do with mental health, which I think is kind of interesting. That was something that came out of this study. But broadly speaking, they want to see whether there's relationships between health markers and the chemicals and socioeconomic context. So where they can actually go in and do social interventions to do with public health and address problems. And so we created a map where there was potential mental health within the community using the data we had. And then they felt it would be very useful for hospital management and decision making in terms of how much chemicals are used in hospitals. So, in conclusion, I know I went through that last part quite quickly. Global AMR will only be solved if we integrate all of the different elements of the One Health spectrum in our solutions. If you only study one, it really doesn't help us much. We're pretty confident the environmental spread of AMR is dominated by environmental factors, more often than not water, but also food contaminated by poor water. But AMR itself is transmitted on a very local scale. So, and that doesn't necessarily directly relate to what you find in the environment right where you are. Latin America, broadly speaking, and this is my observation, really doesn't have adequate investment. That's no one's fault here. But it's a reality. And so there's a desperate need, and I know in the United Nations there's a strong desire to try and promote more integrated surveillance in Latin America. And a project that I'm very much in the back room, Julian is more involved in, called Panacea, that's trying to do this. And what we can do with this integrated surveillance, we want to identify causes, sources, and types of AMR. In other words, we want to create that link, and if we can find that link, then we can find where to break it, so that we can not have that stream of activity occurring. We can answer specific healthcare questions, like the question I made to do with mental health. But we also can answer other things. But you've got to provide data to the people who are your client, and your client, in this case, for human healthcare, are the doctors and the healthcare workers that work in the community. If you don't provide them data they can use, what you've done is really of no great value. It's just an academic exercise. And finally, what we want to do is build a lot of this type of activity into national action plans. Right now, national action plan, most countries in the world have got a national action plan to do with AMR. 73% of the national action plans that exist in the world, in the countries, do not include environmental factors. So they have no hope in their national action plan of actually addressing AMR. Because if they don't consider the environment, they're not considering 60% of the potential cause of drug resistance they find in the country in which they live. And so what we're doing, and this is what I do, this is so almost like I used to do research. I still do research a little bit, but mostly I'm out evangelizing, talking to people about the importance of environmental AMR. But I also talk to physicians, I talk to engineers, I talk to people in the food industry. And I talk to a lot of people trying to motivate people thinking more holistically and more One Health in their solutions. So I thank you. And if there's any questions, I'll answer them.
Thank you very much, David, for this very interesting talk, an example of this integrated surveillance and how to do it. So if you have questions, please raise your hands. Okay.
Thank you. Very interesting talk. I don't know if you know that in Mexico, the biggest wastewater treatment plant that is near Mexico City treats about 35 cubic meters per second. It's the third largest plant in the world. 6 years ago, when the plant is up to be the startup of the plant, I was invited by the in a meeting. This meeting was called by the National Institute of Health, and they were very worried because they said at that time, and I was invited as an engineer of wastewater treatment plant. And they see that the plant is a fabric of MRA. A wastewater treatment plant is very bad for health. The physicians say that. And I have to argue what are the benefits of wastewater treatment against that we don't know what happened. And they said, for 100 years, wastewater is used in this region where the wastewater treatment plant is. Wastewater is used to cultivate crops, different crops. The problem is pathogens that are in the non-treated wastewater. And now, with the wastewater treatment plant, the problem is you remove pathogens, but you produce other bugs that potentially they are worse than the original pathogens. So there is a controversy very huge with this plant that works very well from a technical point of view, but a lot of controversy about this. So the problem is more complex.
No,
I only treat the wastewater.
You've got an excellent question, and I've got I think a number of comments.
The question is, what do you say when they say that the wastewater treatment plants are fabric of potentially harmful microorganisms?
Okay. First, their belief of that is not right. It's incorrect. And so our job is to try and educate them into understanding that wastewater treatment plants are not perfect, but the spread of resistant disease is infinitely worse if you don't have a treatment plant. And one of the key things, and what you said was that, say, well, their treatment plants are creating these new pathogens. It's simply wrong. It's not true. And there's huge amounts of evidence to say that's not true. And so our job as engineers is to try and present them with information, this is the people in the healthcare side, of evidence that that isn't true. Now, one of the problems we have is we have a tendency to use and measure things that tell us things that aren't relevant. In other words, if you're doing a metagenome and you got all these genes and all these nasty genes, but it doesn't really mean anything unless it's related to resistant strains. And so what we would do in your case, I know the treatment plant you're talking about. I think it's a tremendous treatment plant. I don't think there's a problem. And so what I would do is I would provide a case, I would provide evidence for them to show them that in fact what's going into the treatment plant, have them tell you, okay, you've told me that there's super resistance, you're talking to them. You've told them the super, okay, let's grow organisms that go before the treatment plant, and let's measure what those organisms are after the treatment plant. I would guarantee to you that there's 100 to 1,000 times fewer of those organisms. And there's also no new resistance. New resistance evolution happens in a more nebulous way in the environment. Waste treatment plants aren't incubators for new waste resistance. It's possible, like anywhere else, it can happen. But we've studied somewhere between 20 and 30 treatment plants in different countries. And only in 2 cases did we find any new development of resistance in the treatment plant. And those were both, one was in Sri Lanka and one was in India. And those treatment plants were operated terribly. Their settlers didn't work. And so they came away, people came away, and this was sensational. This was the one in Sri Lanka. It was headline news. This superbugs being created by treatment plant. Well, this treatment plant didn't have a settler that worked. The treatment plant was just a concentration of all the sewage that was going through the treatment plant, but the treatment plant wasn't doing anything. So people concluded that the treatment plant was creating resistance, and in fact it wasn't. It was a treatment plant that wasn't functioning. Now in that Mexico City treatment plant, it's very well run. And so if I wanted to make my argument to your health official, I would just tell them, okay, carbapenem resistant Enterobacter, vancomycin resistant. You could basically choose those and just culture the organisms in and culture the organisms out using the methods that the medical people use, and I think they'd be pretty convinced that it's not a problem.
Yes, it's true, because the physicians that were supporting that belief were using an article that produced by Harvard University.
Yeah, doesn't mean they know what they're doing.
And when I read the paper,
yeah.
I don't know what type of treatment, etc. But they say this is Harvard that said that.
Yeah.
So very, very close, because they were physicians. So we were like engineers to explain how the wastewater treatment improves health, not worse health.
Well, you're completely correct. And one of the things that is there was a lot of myths to do with wastewater treatment plants that weren't true. And if you look at the literature over the past 15 years, you'll see the literature prior to about 5 years ago would say wastewater treatment plants are horrible. There's dens of new resistance formation. You look at stuff now, especially the last 2 years, that's reversed. So Harvard might have done a great study, was wrong, but it was a great study in 2011 when people didn't know how to do the studies correctly. Now we know how to do them right. And we can go back to those people and say, I'm doing some work with the Chinese Academy of Science right now, where we're trying to look at what people believe about AMR and the environment based on looking at using artificial intelligence to look at what people believed in the 2003 to 2010, 2010 to 2015, 2015, 2000. So we were trying to look at how people changed the way they thought as mirrored by what they do in academic and government and other related activities. And the last 3 years things have changed a lot. And the fact that this report exists, if I was being, I'm sort of biased because I was involved in writing it, but if you show this, we've got a simple version of this report. This report's about 80 pages. We've got a simple one that's 12 pages long. And it summarizes, it's called AMR in the Environment for Policymakers. So it's very small, it's put into simple terms, and it's put into terms that somebody who's maybe running a health trust could understand. But what you said to them is absolutely true. I completely understand and completely agree. But things have changed a lot.
Thank you, David. Thank you, Herman, for the question. We have a comment from online, people watching us online. It's from Maria Inés Sato. She said that the high risk of waterborne AMR exposure in Brazil could be consequence of the high indiscriminate use of antibiotics. I don't know if you have a point of view about that.
Oh, I completely agree. Yeah, yeah. I mean, I could talk about different countries because we've done a lot of work around Europe. And in Southern Europe, particularly Spain and Italy, antibiotics are used at a higher level than other parts of Europe. And they're also warmer climates. But one of the big differences there is that they use more antibiotics. They also use them not in Spain and Italy as much, but in Brazil and in, say, India and in China, they're used for things that are not therapeutic. They're used for growth promotion in animal agriculture. They're used for other different purposes. But so yes, that's one of the reasons, and also so it may not just be pollution, which may exist. It could very well be different behavior. So, and that's once again, it gets back to One Health solution. So part of the One Health solution is you use antibiotics more prudently. You don't use them indiscriminately for some purpose in the environment that's not really needed. And if you've got a sick patient, there's a move away of using multi-spectrum antibiotics. In other words, these are ones that kill everything everywhere. They're trying to come up with more tailored, specific antibiotics to particular diseases. Because if you use a multi-spectrum antibiotic, it makes you an organism resistant to everything. And so there's a very strong push in new drug discovery towards sort of tailored boutique antibiotics that will kill a very specific disease. And that will then mean that you're not going to create, it's sort of like collateral damage in military. If you bomb somebody, you kill everybody around the bomb site. If you've got something that's a very specific, insightful weapon that just hits the point, then you don't create all this collateral damage. And so it's the Brazilian, I'm pretty sure that's true.
Thank you, David. Do you have another? We have another question over here.
I have two questions. One was how we can identify that we have the antimicrobial resistance. I mean, we have to do a specific study in laboratories, or if we can identify with any study on laboratories.
Antimicrobial resistance studies don't require you to do anything particularly different. So if you are studying microbiology, you say you're growing microorganisms on plates. Well, you grow organisms on plates, but some of those plates you add an antibiotic. And if the organism grows and the antibiotic's there, you know they're resistant. It wasn't a lot of extra work. We also know we've probably got an archive of thousands of genes that are associated with different forms of resistance. It's just like doing any other sort of genetic categorization. We know the genes to look for. And so including AMR in what you do is really easy. And so we do it all the time. So we've got a study on some topic for my example to do with Cuba. Our study was on metal pollution. But we also know that there's potential association between metal pollution and AMR. So we just grew a couple of plates. And we quantified, used qPCR on a couple of genes. Okay. And it was just there. It was so blatantly obvious that AMR was developing in certain places and it was different. And then we invested more. So
okay,
it's not hard. It's pretty easy. And I think the thing is it takes a bit of experience to know which to look for, which type of resistance. But that's one of the things that we're doing right now at the UN is we're trying to come up with a recipe book, a guide, so that if you're interested in this resistance, you use this method. If you want this resistant, you want this method.
Okay. And the second one is, it exists a study to say that these pathogens can be transmitted to others, even if they're pregnant and they generic can transmit?
So I don't understand the question.
Así que, o sea que sí se puede transmitir ya sea por genes, o sea, genéticamente esos patógenos se pueden transmitir. O sea, de una persona que no tiene
Is there any evidence that this antimicrobial resistance can be transmitted from one person to another, or to a pregnant woman to child, something like that?
I don't think that you would see resistance transmitted between a mother and a child. I don't have any evidence. I don't think that would be possible. Having it transmitted from one person to another, that happens all the time, and we can quantify that. So what we have learned, though, in recent times is that we used to think that it was easy to transmit. In other words, I've got a resistant pathogen in my gut, and I walk by the professor here, and he gets it. It doesn't work that way. It's actually more difficult than that. But if I have food that's got resistance on it, a resistant bacteria, you eat that, I eat that. Or I eat it, get it on my hands, contaminate your food, you get it. That happens. And I think there's a lot of evidence of that. And so that's where we're trying to understand. That's part of the reason why we want to understand things in these informal and poor settings, where they don't really have any healthcare protection. So how much does it happen between one person and another? And it doesn't seem to, it more seems to happen by one person contaminating food, and that food is then getting eaten by somebody else. So it's not a direct thing always, unless you kiss them, but it's indirect, but it's pretty close.
Thank you for the answer. We have more comments online. Mauro Miguel Garcia Enriquez, congrats you and thanks for the presentation. We have a comment. It says that, hi David, I'm Mariano from Santiago. What is the probability of cross-resistance occurring between microorganisms in the discharge of wastewater treatment plant?
Of co-resistance.
Cross-resistance.
Cross-resistance.
Yeah.
Oh, I understand. It can happen. Yeah, we don't see as, this is just based on my own experience. I can't talk about everything, but what we see in treatment plants often is selection of resistance rather than new development of resistance. It's conditions within the treatment plant that selects for a particular type of resistance. Now, the issue of cross-resistance is very common in the environment. And there's an engineering reason for that. And this is the reason why, and that's why I have to be a little bit less certain about this. But a lot of these resistance exchange phenomena, they take time. They're not like that. They're not sudden. Treatment plants, if you look at the hydraulic retention time in a treatment plant, it can be 4 to 8 hours. 4 to 8 hours is not a lot of time for evolution to occur. And even at the bacterial scale. Now, there are some processes in treatment plants where you can potentially have more time, say in a sludge recycle line. You can get more contact time there. But we did a study, this was many years ago, 15 years ago, looking at the effect of trace levels of antibiotics in animal lagoons in livestock farms in the United States. And we found that we could get resistance, cross-resistance to occur, but it took 57 days. That was when we first started seeing it to appear. And that was at moderately high concentrations of the antibiotic. So, I can't answer this question specifically, but my impression is in treatment plants there probably isn't enough time for it to significantly happen in the treatment plant. So if you see cross-resistance, it's more than likely cross-resistance that developed along the path, or it's more likely to occur after the treatment plant in the environment because there simply isn't enough time.
Thank you for the presentation, Doctor. And I have a question. And does Panacea have any influence with the government of Latin countries to promote the wastewater surveillance?
Do I have influence? Or do you, well, sorry, are you asking, well, how can we create influence? Or I didn't quite understand the first part. I understand the general principle. I can make sort of a general comment. As in the places in the world where there isn't integrated surveillance, it's usually in places where the government doesn't see any immediate value in the surveillance. And part of the reason why we did the World Economic Forum study was to try and put dollars and cents to resistant disease. So we wanted to say, okay, if you don't have resistance in your national action plan, resistance prevention in your government's national action plan, we're now talking to the Minister of Health, this is how much it will cost you. This is how much more expensive your healthcare system will be if you don't handle the problem correctly. And one of the side benefits of what I do is I sometimes talk to government people at moderately high levels, and that's what I say. Okay, let's get a napkin out, and let's do some calculations about how much, if you don't do anything, will cost you. And one of the things I think found is some politicians are actually altruistic, and they do things for the right reasons. But I haven't found a single politician that isn't influenced by money. I'm not talking about bribing or anything like that, but if you can show somebody that you can save 10 million, or say $100 million dollars per year, if you prevent resistance at a certain level, I'd be shocked if they wouldn't act. And so it's that type of thinking we should be having, is that we've got to try and put you got to put yourself in their shoes. Can you think? We're all here. We're all pretty smart. Yeah, we're all. Some people are a bit smarter than others, but the bottom line is we're all pretty smart here. And the politicians, they're also smart. And you've got to put yourself in their shoes, if that makes sense in terms, that's English. So you put yourself into the way they think. And then you think about what you need to tell them to make them make a decision, because people don't change their mind very easily. So what you want to do is give them all the evidence, which is biased, to try and make them think a bit differently about what they do. And so we say, okay, if you don't do anything, and this is your economy, and this is your workforce, and this is the cost of this and the cost of that, and they listen. Now, you sometimes have trouble getting in the room because they've dismissed you before you're even in the room. But that's what we're trying to do right now, and as we're trying to, I said we wrote this report, which we're really proud of. But the more important thing from the report was the World Health Organization, UN Environment Agency, and the different agencies agreed that the environmental relevant part is important. And that creates a massive amount of leverage in terms of the politician you might talk to, or the minister of whatever, is that now we've got validation. We've got validation. You've got this. We've got the policy guide and so we're in the process of now. So we've come up with the general framework. We've convinced the different agencies of the UN that all these different parts of our world and our life has to be considered in the solution. We're writing a guide that tells you, if you want to include resistance in your study, how to do it. And the guide is gauged relative to the technological capacity of a place. So the guide for one part of the world based on this level of capacity would be this. if you've got that capacity or if you've got that capacity, the guide tells you that. And the next step, though, is, okay, we've got these tools. How do we actually get people using them? And that's your question. That's your question. And that's what we're trying to do now. So the guide is being written, the evidence has been provided, and now we're actually saying, oh, how do we go about getting people in positions of influence to understand the problem? Part of that is explaining things in a simple way. Academics have a terrible time doing that, because academics have a tendency to try and make things really complicated. But you want to try and compress it down into pieces of information that can be useful. And in fact, and I say this, I've only seen it work once, where we've been able to convince people of something. But we now have better evidence, and there's a bit of momentum going on right now. So we're hopeful. But I think this is a particular problem in Latin America. Yeah. Is that you've got governments, I mean, this is very broadly speaking. Generally speaking, governments haven't been very responsive to COVID monitoring, for example. There's been wastewater monitoring to do with COVID. Some really useful evidence and data has been generated. I was involved in work in Paraguay, which was really successful in terms of what we did. It was done by our colleagues in Paraguay, but we worked with them in setting up an equivalent network to the ones that we had in the UK. But in that country, there were people within the healthcare system that were willing to listen. But it's the only place that I found it. And the question is, how do you get those politicians to prioritize this type of thing? And that's money.
I will ask something related, that in the case like country like Mexico, that maybe there is no data available about costs.
Yeah.
I don't know how to translate or put in the Mexican politician's shoes. Maybe in translating in votes for next elections, I really don't know, because I think we did work about COVID in wastewater, and so similar result that you said in Paraguay. People even don't listen, and not only here, also in Mexico City and other states. We did pilots in several states in the country. So it's kind of tricky, I don't know, in some places like Latin America, how to translate or put it in numbers that politician or decision maker cares about it.
I mean, I realize that's a problem. I think we, though, and this is where I'm critical of us, is I think when it came to COVID, we didn't provide the politicians and the healthcare people the information they needed. I think we certainly know in the UK, I don't know Mexico, but in the UK, people who were doing the wastewater work didn't sell what they could do right. It was, in other words, they sold wastewater monitoring as providing you an early warning system to prevent cases. We had this 3 to 5 day lag based on exposure before you saw symptoms. So you now had 3 to 5 days that you could get a signal in wastewater that you could tell somebody in the healthcare system that there's a case. I think that telling them that was the biggest mistake we could possibly say, because it's just too inaccurate. And the inaccuracy is not in the COVID analysis. We can measure it exactly in the sewage. Our data in the sewage is really accurate. The problem is it's translating it into case data. And it's just not black and white. And there's too big an error bars. So what we did was, we said by saying we could create an early warning system, I mean, this is a long answer to a different question, but we created an early warning system. And in fact, it's like false advertising. We said this is what we were going to do, but so we shouldn't have focused so much on that. What we have found with wastewater is not an early warning system. We found wastewater monitoring incredibly good for finding new variants. where you do sequencing of samples. And that is useful. And I can sell that to somebody in the government, because if they can know ahead that they've got a particular type of new variant that isn't, it gives you a flag that maybe in a month that variant could develop. So I think it wasn't false advertising. I think it was overly ambitious advertising, and it created. And this is exactly what happened in the UK, in England especially. That I wasn't actually, to be honest, I was against this principle, saying we could use it as an early warning system. At the very beginning, I thought it would work. As soon as I had a little bit of data, I realized it wasn't going to work. And so I was promoting that we should be using it for surveillance and variant screening and that sort of thing. So we've got to be a bit, and we also finally, in the final part of this long answer to a short question, is that we've got to provide the data in a form that the people can use. In other words, healthcare decision maker needs data in a form that means something to them. And if you provide a metagenome or virome, I don't even understand it. And how are they going to understand it? So we have to consolidate and compress the information we generate into something that's understandable. And so when it comes to this, I think we misadvertised what we were doing. I think we underplayed the things that were of great value, and we messed up. We didn't handle it right. And in some countries, though, it's fortunate. So in Wales and in Scotland and in the Netherlands and in Germany and Turkey, there's countries that are continuing on to do it, and where they've been able to make this translation to longer-term strategic value rather than immediate value. So.
Thank you so much, David. Thank you, everyone, for your having here, and thank you, the person online. So, if you have any question, David will be here during the week. So he's in building number 1 in the, how do you say that, in the meeting room. So he will be available. Thank you, everyone.